You feel completely fine. That is the problem. You ate the mushroom hours ago, maybe yesterday. You're not dizzy, not nauseous, not in pain. Your body is giving you zero signals that anything has happened. And while you go about your day feeling entirely normal, your liver is being dismantled at the cellular level with a precision that no pharmaceutical compound has ever matched. By the time symptoms arrive, the damage is already done. The window to intervene has already closed. And in documented cases around the world, that window is measured in hours from the moment of ingestion, not from the moment symptoms appear, from the moment of ingestion. When you feel nothing at all, that is not a horror story. That is the documented, peer-reviewed, clinically observed biology of one specific mushroom. And the reason it kills people is not that it is some exotic jungle species or a rare find in restricted terrain. It looks like a mushroom you would buy in a grocery store. It grows in parks and backyards and forests within walking distance of cities on every inhabited continent except Antarctica. Every year, according to mycotoxicology literature reviewed by the World Health Organization, this single species accounts for roughly 90% of all mushroom-related deaths worldwide. Not every toxic mushroom combined. One species. The death cap, Amanita phalloides. And the reason it kills, the actual biological mechanism, the thing no one in your life has ever explained to you, is one of the most precise, merciless, and counterintuitive processes in all of toxicology. The symptom delay is not a side effect or an anomaly in the data. It is a structural feature of how the primary toxin operates at the molecular level. Once you understand what is actually happening inside the body during those silent hours, you will never look at a foraged mushroom the same way again. If you enjoy understanding how the world actually works beneath the surface, subscribe. It is the best way to support this work. Drop in the comments what country you are watching from. This script draws on research from the New England Journal of Medicine, The Lancet, and the Journal of the American Medical Association, alongside toxicological databases maintained by the World Health Organization. Now back to it. Here's the first thing you need to understand. The death cap does not contain one toxin. It contains several chemical compounds, including phallotoxins and virotoxins. But only one class of compounds is responsible for the killing. They are called amatoxins. And the most lethal member of that class is alpha-amanitin. Alpha-amanitin does something that almost no other poison on Earth does at this level of specificity. It does not attack your nervous system. It does not stop your heart. It does not cause immediate pain, bleeding, or spasm. It goes directly for the thing that makes every single cell in your body capable of doing anything at all. Plain verdict. Alpha-amanitin stops your cells from reading their own instructions. Every cell in your body runs on a continuous process of reading genetic instructions and producing proteins based on those instructions. Proteins are not just structural components, they are the machinery. They are the enzymes that run your metabolism, the repair crews that fix damaged tissue, the signaling molecules that coordinate organ function. Everything your body does requires proteins, and proteins require a functional molecular machine called RNA polymerase II to produce them. Alpha-amanitin binds to RNA polymerase II and locks it permanently in place. The machine cannot move. The cell cannot build new proteins. Without new proteins, the cell cannot maintain itself, cannot repair damage, cannot continue functioning. It does not explode. It simply stops being able to sustain itself, and then it collapses from within. Here is why that is insane. Most poisons work fast because they target systems your body runs continuously and in real time. Your nerve signals, your breathing muscles, your cardiac rhythm. Alpha-amanitin targets something deeper than any of those. It destroys the factory that builds the components that keep all of those systems running. It is not cutting the power, it is disabling the manufacturing capacity that produces every replacement part in the entire facility. And because cells do not die the moment protein production stops, they coast on the proteins already present for hours, the body has no immediate alarm to send, no spike of pain, no nausea, no warning signal of any kind. Think of it like this. Your cells are running a factory. The factory has machines that read blueprints and produce components. Alpha-amanitin does not burn the factory. It destroys only the blueprint reading machines. For hours, the parts already in inventory keep the factory running. The lights stay on, the conveyor belts keep moving, the product keeps coming off the line. From the outside, from every external measurement, everything looks completely normal, but nothing new is being manufactured. And when existing components wear out, and they always wear out, there's nothing to replace them. The factory does not explode. It fails silently, system by system, until it cannot run at all. The technical name for the machine alpha-amanitin destroys is RNA polymerase II. It is the enzyme responsible for transcribing DNA into messenger RNA, the intermediate step between a genetic instruction encoded in your chromosomes and a finished functional protein. Alpha-amanitin binds to a specific site on this enzyme with exceptional chemical affinity and physically locks it in place. The enzyme cannot advance along the DNA strand. Transcription stops. the clock starts. Now here's the piece that explains the silence and the geography of how death cap poisoning actually kills. Your liver cells have the highest concentration of RNA polymerase II activity in the human body. Not because the liver is special in a vague general sense, but because the liver performs an extraordinary number of concurrent biochemical tasks, all of which require continuous protein production to sustain detoxification, glucose regulation, bile production, synthesis of clotting factors, processing of waste products from cellular metabolism. All of these run simultaneously, and all of them are protein dependent. The liver is the busiest factory in the body. An alpha-amanitin, once absorbed through the gut wall into the bloodstream, is delivered directly to the liver through the portal vein, the blood vessel that routes everything absorbed from the intestines straight to the liver before anywhere else in the body receives it. The liver gets the full dose first, and then something happens that turns a single exposure into an extended assault. After the liver processes alpha-amanitin, the toxin is excreted into bile, travels to the small intestine, gets reabsorbed through the intestinal wall, and is sent straight back to the liver through the portal vein again. This cycle, called enterohepatic recirculation, means the liver is not hit once. It is hit repeatedly by the same molecules, cycling through the hepatic blood supply again and again across many hours. The same toxin, the same binding event, the same RNA polymerase II shut down in each new hepatocyte it reaches. The exposure does not end when absorption from the gut is complete. It continues for as long as the recycling loop runs. This is why the symptom delay is not a gap in the biology. It is the biology working exactly as its molecular logic dictates. Liver cells are dying on a schedule. The first wave of cell death has not yet reached the threshold required to trigger systemic symptoms. The patient feels nothing because the visible painful damage, the kind that produces jaundice and organ failure, requires a critical mass of cellular collapse that takes time to accumulate, even as the recycling loop continues to deliver additional toxin to a liver already under assault. The clinical timeline in documented human poisoning cases is remarkably consistent. Symptoms of gastrointestinal distress, nausea, vomiting, abdominal cramping, watery diarrhea appear between 6 and 24 hours after ingestion. The majority of cases cluster around 8 to 12 hours. Then those initial gut symptoms resolve. The patient feels better, often significantly better. In medical literature, this period has a formal name. It is called the honeymoon phase. Physicians who have treated amatoxin poisoning describe it as the most dangerous point in the entire clinical course, not because of what is happening symptomatically, but because of what the apparent recovery leads patients and families to believe. That improvement is not the body healing. It is the gastrointestinal tract finishing its acute inflammatory response while the liver continues to sustain compounding damage from a toxin cycling through it that the gut no longer registers. The biological front line has moved upstream, and without blood tests measuring alanine aminotransferase and aspartate aminotransferase, the liver enzymes that rise sharply when hepatocytes are dying, there is nothing from symptoms alone to indicate that anything is continuing to go wrong. The patient feels recovered. The liver is failing. Now here is a detail that makes this worse. The honeymoon phase does not just delay treatment, it actively discourages it. Patients who seek medical attention early, during the gastrointestinal phase, are sometimes discharged after initial assessment when symptoms appear to resolve. Without a high index of clinical suspicion and a history of mushroom ingestion, an emergency physician seeing a patient who reports feeling better may not immediately order the full liver function panel that would reveal the damage already underway. Several documented cases in the toxicological literature describe patients discharged during the apparent recovery phase who returned 48 hours later in hepatic failure. The biology of the honeymoon is not passive. It actively mimics recovery in a way that interacts with clinical decision-making processes. This is why toxicologists and emergency physicians who specialize in poisoning cases treat any suspected amatoxin ingestion as a liver failure in progress, regardless of how the patient presents. The absence of symptoms is not reassurance. It is part of the mechanism. It is also, from the perspective of anyone trying to understand how a natural molecule could be designed more precisely to evade medical intervention, a near-perfect engineering solution. Between 48 and 72 hours post-ingestion, liver failure becomes clinically undeniable. Jaundice appears as bilirubin, a waste product the failing liver can no longer process, accumulates in the bloodstream and tissues. Coagulopathy develops. The blood loses its ability to clot because clotting factors are proteins. Proteins require functional hepatocytes to produce, and those cells are no longer functional in sufficient numbers. Kidney injury follows because the kidneys are managing filtration loads the liver can no longer handle. In severe cases, hepatic encephalopathy develops as toxins accumulating in the blood, cross the blood-brain barrier, and impair neurological function. Confusion, disorientation, coma, the sequence moves in one direction. The liver, the organ the body uses to protect itself from chemical harm, is the organ that is destroyed first and most completely. There's a clinical irony in that which toxicologists note without embellishment. The estimated median lethal dose for alpha-amanitin in humans is roughly 1/10 of a milligram per kilogram of body weight. A single death cap mushroom contains between 5 and 15 milligrams of alpha-amanitin, depending on the specimen and its maturity. An average adult weighs roughly 70 kilograms. Half a mushroom, depending on concentration, may be sufficient. The math produces a number that clinicians in toxicology do not describe as a margin. They describe it as the absence of one. Here is something that should stop you completely. The death cap does not taste like poison. In multiple documented cases published in poison control literature across Europe and North America, victims who survived described the taste as pleasant. In several recorded fatality clusters, families cooked the mushroom carefully, shared a meal, found it good, and went to bed feeling fine. The gastrointestinal symptoms did not arrive until the following morning. In at least 2 documented case series from California, immigrants from Laos and Southeast Asia mistook the death cap for Volvariella volvacea, the paddy straw mushroom widely consumed throughout Southeast Asia, which grows with a similar partial veil and surface profile. The visual similarity was sufficient. The biology was not forgiving. There is no smell warning, no bitter or acrid taste that triggers aversion, no visible marking that distinguishes it reliably from edible species in the field. The death cap is not advertising its toxicity because its ecology does not require it to. It reproduces through spore dispersal. Whether individual fruiting bodies are consumed by animals has no bearing on the reproductive success of the fungus. The mushroom did not evolve to warn you. Alpha-amanitin is not a defensive chemical in any active behavioral sense. It is a metabolic byproduct whose lethality to vertebrates is, from the mushroom's ecological perspective, entirely incidental. The global distribution of Amanita phalloides has expanded significantly across the last century, and the mechanism of that expansion is specific and documented. The death cap is native to Europe, where it evolved in mycorrhizal symbiosis with oak, beech, chestnut, and pine trees. This is a relationship in which the mushroom's mycelial network exchanges minerals and water with tree roots in a mutual biological dependency. When European tree species were transplanted to other continents for timber production, urban landscaping, and ornamental planting, the death cap traveled in the soil attached to root systems. It is now established across North America, South America, Australia, New Zealand, and parts of Africa and Asia. Populations with no prior cultural exposure to the species, no generational knowledge of its appearance or danger, are encountering it as a new hazard in terrain where it did not exist before. The California death cap fatalities that received significant clinical documentation beginning in the 2010s reflect exactly this pattern. Communities with deep foraging traditions from regions where Amanita phalloides is not native arrived in a landscape where the death cap had established itself in city parks and suburban woodland. The mushroom looked familiar enough to harvest. It tasted fine. The enterohepatic recycling loop ran its course. There's currently no antidote that neutralizes alpha-amanitin after ingestion. Treatment is supportive and depends entirely on how early clinical presentation occurs. Activated charcoal administered within a few hours of ingestion can interrupt the enterohepatic recycling loop by binding the toxin in the gut before reabsorption, reducing total hepatic exposure significantly. Aggressive intravenous fluid support maintains kidney function. Continuous liver enzyme monitoring tracks the rate of hepatic damage. Silibinin, a compound derived from milk thistle, has demonstrated in European clinical trials the ability to disrupt hepatic uptake of amatoxins by competing for the transport protein that carries alpha-amanitin into liver cells. It is used in European centers as an off-label intervention and has been associated with improved outcomes in early presentations. In the United States, it has been administered under compassionate use protocols in several documented cases. In patients where hepatic failure progresses past the threshold beyond which regeneration is not possible, where cumulative cell death has exceeded what the liver can recover from, the only intervention that changes the outcome is emergency liver transplantation. The timing window for that decision is narrow. It requires rapid clinical assessment of the trajectory of liver enzyme elevation, the patient's coagulation status, and the availability of a compatible donor organ within a system that allocates organs under significant logistical constraints. Survival in severe cases correlates directly with speed of initial presentation. Every hour between ingestion and hospital contact, while the recycling loop continues running uninterrupted, narrows the clinical options that remain available. Here is the piece that closes everything you have just heard. Toxicologists classify poisons along 2 axes, mechanism and onset. The death cap sits at an intersection that is genuinely rare in the toxicological literature, a delayed onset hepatotoxin with an active recycling mechanism that amplifies total exposure over time. Most hepatotoxins either produce early symptoms that drive rapid hospital presentation or are toxic at doses that also generate other acute systemic effects, compelling immediate medical contact. Alpha-amanitin does neither. The enterohepatic recirculation both extends total hepatic exposure and simultaneously maintains the delay in symptomatic onset. It is not that the toxin acts slowly in its molecular mechanism. RNA polymerase II inhibition happens immediately at the cellular level. It is that the consequences of that inhibition accumulate silently across a period that corresponds almost precisely to the interval during which clinical intervention would be most effective. The delay and the lethality are not separate features. They are the same feature expressed through the same biology. The mechanism that makes alpha-amanitin lethal is the mechanism that makes it invisible until the clock has run. The rule this produces is simple and has no exceptions. If you cannot identify a wild mushroom with complete, verified certainty, not high confidence, not reasonable likelihood, complete certainty confirmed through multiple distinguishing morphological features, cross-referenced against verified reference material, or confirmed in person by a trained mycologist, you do not eat it. Not because most wild mushrooms are dangerous. The majority of species encountered in temperate forest are not. Because the one that is dangerous does it in the specific way that shows you nothing until the window is closed. Heinrich Wieland and his colleagues at the University of Munich first isolated and chemically characterized the amatoxins in the 1950s. The precise binding mechanism, inhibition of RNA polymerase II, was not resolved at the molecular level until the 1970s, when the structure of the enzyme toxin complex was fully characterized. Alpha-amanitin had been killing people for all of recorded human history before anyone understood what it was doing inside a cell. Hippocrates documented mushroom poisoning in ancient Greece. Roman historical records contain accounts consistent with amatoxin poisoning, including the possible death of the Emperor Claudius in 54 AD from a meal of mushrooms, an account historians and forensic toxicologists have revisited repeatedly without consensus, but one that is not inconsistent with the documented clinical timeline of Amanita phalloides ingestion. The molecule has been operating by the same mechanism for longer than recorded history. The understanding arrived 2,000 years later, and the understanding changes nothing about what the mushroom does.